A very good morning, friends. So today's topic of discussion is molecular basis of cancer. In previous video, we have just discussed how the proto-oncogenes develop into oncogene. If you don't watch the video, please go through that video so that you can easily understand the concept. So today we will discuss the role of tumor suppressor gene in the development of cancer. So what is this tumor suppressor gene? These are nothing but these are the normal genes. These are the normal genes present in all healthy cells. Okay. So these are nothing but these are the normal cells. Genes which are present in all healthy cells, and these are the growth inhibitory gene. Growth inhibitory gene. As the name suggests, tumor suppress. Suppress means inhibition something. So these genes will growth inhibitory gene, as these will inhibit the growth of cell. Now, the tumor suppressor gene classification. How you will classify the tumor suppressor gene? So classification includes, first is that of RB gene. What is RB gene? RB gene is also known as retinoblastoma gene. Retinoblastoma gene. First category is RB gene. Second is P53 gene. And third is TGF beta. What is TGF beta? TGF beta is tumor growth factor beta. Okay. And next category is that of ACP gene. And fourth category is others. In others, we just included mainly two kind of tumors gene, that is BRCA1 and BRCA2. And these BRCA1 and BRCA2 genes, when these are mutated, then they will cause in human is that of breast cancer. Clear everyone? So these are tumor suppressor gene. And other is that of WT1 and WT2 gene. And these WT1 and WT2 genes are associated with Wilms tumor in human being. And this Wilms tumor is a childhood tumor of kidney. So this is the classification of tumor suppressor gene. So next we will just discuss how the mutation occurred in this tumor suppressor gene and how these mutations will develop a normal healthy cell, transform it into a cancer cell. What happened on this tumor suppressor gene? Okay. So in this video, first we will study the RB gene. What happened with the RB gene? And in next video, we will study the P53 gene. So RB gene. RB gene is what? This is the retinoblastoma gene. And it is normally present. Where it is present? It is normally present on chromosome number 30. Okay. And the important thing is that the activity of this RB gene, depends on its phosphorylation state. What does it mean? Depends on its phosphorylation state. It means whether you will say whether RB is active or RB is inactive. Its activity and inactivity depends on its phosphorylated state. Whether RB is phosphorylated or whether it is dephosphorylated. Now we will see what happened in the normal cell. Suppose cell wants to divide. Cell start division. Cell start to divide. Then what will happen? If cell start to divide, as we have just studied in previous lecture, when cell start divide, then there will become a growth factor. Okay? Then growth factor like platelet-derived growth factor, epidermal growth factor, etc. There are so many kinds of growth factors which are required for cell division. So these growth factor will come, and then these growth factors will attach with the growth receptor. So growth factor plus growth receptor complex will form. Okay. So when these growth factor will come, these will combine with the growth receptor and then it will form a complex. So what happened when this growth factor, growth receptor complex will form, then what it will do? It will just activate some kind of cell regulatory proteins. What it will do? It will activate, this complex will activate cyclins and CDKs. What are these cyclins and CDKs? These cyclins and CDKs are nothing but these are cell cycle regulatory proteins. So these cyclins and CDKs are required for the normal cell cycle. What is the full form of CDK? CDK is cyclin dependent kinase. Clear? As kinase, as in whole biochemistry, wherever the term kinase comes, that means it will phosphorylate something. So as it's a kinase, that means it will phosphorylate something. Then it will phosphorylate the RBG. It will phosphorylate the RBG. Suppose this is the RB gene. This is the RB gene. Okay. And cyclins and CDK is active. And these cyclins and CDKs cause phosphorylation of this RB gene. Clear? And when this RB gene is phosphorylated, then this phosphorylated RB gene is in inactive state. Clear? So when RB is phosphorylated, then it's become in inactive state of RB gene. And when RB gene is in its inactive state, what it will do? It will release transcription factor. It will release transcription factor. And mainly which transcription factor? That is EF2. Okay. So suppose this is RB gene and now phosphorylated RB gene is active. This is RB gene and this is phosphorylated. And this phosphorylated RB gene is active. Then what will it will do? It will release this EF2 factor. And this EF2 is a transcription factor. So what it will do? It will start cell division. Okay. So this phenomenon is occur normally in the cell. Now cell start divide and suppose if cell division complete. Now cell don't want to divide further. Then what will happen? Now cell is saturated and whole of its cell is divided. Then it should be stopped now. Isn't it? Then what will happen? Then when cell division complete. So when cell division complete, then what will happen? Growth inhibitors will come. Because now cell don't want to divide further. So growth inhibitors come. And what these growth inhibitors, as these are inhibitors, so what they will do? They will inhibit inhibitors. So these will inhibit the cyclins and CDKs. Okay. So when cell division complete, then what happened normally? Then growth inhibitor will come. When growth inhibitor will come, then these growth inhibitor will inhibit the cyclins and CDKs. And when cyclins and CDKs are inhibited, is they are capable to do phosphorylation? No. Then RB gene is dephosphorylated. Or you can also say hypophosphorylated. Then RB gene is dephosphorylated. Okay. Then this is your RB gene and it is not phosphorylated. Okay. And when this is dephosphorylated, then this RB gene is in active state. Then this RB gene is active. And when RB gene is active, it forms complex with your EF2. It will form complex with transcription factor EF2. Okay. What will happen? This is your RB gene and now it becomes what? Dephosphorylated. It's not phosphorylated. When it is not phosphorylated, that means it is in inactive state. And when it is inactive state, then it is in active state. When it is dephosphorylated, it is in active state. When it is active state, then this RB gene will form complex. It will form complex with EF2. This is your EF2. Okay. And this RB and EF2 complex is formed. When this complex is formed, then it will tightly hold this EF2. So this complex hold RB is not tightly hold the EF2 and then EF2 is not available for the cell. So when EF2 is not available, then cell division stopped. Then what will happen? Then cell division stop. Okay? Cell division stop. Now cell stop to divide. So this happened. This whole phenomenon happened normally. Now comes the point how the cancer develops if some kind of mutation will occur in tumor suppressor gene. As we have studied previously the type of mutation in proto-oncogenes for conversion of oncogene, similarly in tumor suppressor gene, when it is mutated, suppose mutation occur, then it will cause, what it will cause? It will cause cancer. Now, two type of mutation is most common in tumor suppressor gene. One is that of point mutation and other is that of chromosomal deletion. So, okay. Then point mutation and chromosomal deletion. These two mutation is common in tumor suppressor gene. Now, suppose point mutation occur in RB gene. Now that normal RB gene comes in its mutated form. What happened? Point mutation in RB gene. Okay. When point mutation occur in RB gene, then RB is in its mutated form. Mutated RB. And being a mutated RB, what happened with this mutated form of RB? It is permanently in its which state? Permanently in its inactive state. Okay, inactive state. And you have studied in normal cell, when RB is permanently inactive, then what happens? There will be permanent release of EF2 factor, because it is EF2 factor. What happened? Because when RB is mutated, then this mutated form of RB is in its permanent inactive state. And when it's in inactive state, then it is not capable to hold or form a complex with EF2. So when it is not capable to form complex or it is not capable to hold the EF2, then this EF2 is continuously released in the cell. So being a transcription factor, when this EF2 is continuously released in the cell, what will happen? Cell divide in uncontrolled fashion. Cell division in uncontrolled fashion. So when uncontrolled division occur in cell, then it will cause what? It is develops cancer, because you know from previous video, cancer is nothing but is an uncontrolled abnormal growth of cell. Clear? So this is how, and this is the mechanism how the RB gene is associated with the development of cancer. Now, example comes, what kind of cancer occur in human beings? For example, when RB gene is mutated, then there is a development of retinoblastoma tumor. It is a kind of eye tumor. Retinoblastoma is nothing but it's a eye tumor when there is a protrusion of eyeball occurs, and it occurs mainly in childhood, less than 5 years of age. Okay, it mainly a childhood age tumor and mostly affect children less than 5 years of age. So clear everyone how the tumor suppressor gene is associated in basically RB gene is associated with the development of cancer. So that's all for today's session. In next session we will study the P53 gene, how it is associated with cancer. Thank you everyone.